Good morning, friends. In this class today, we are going to discuss about what is cell theory, prokaryotes, and eukaryotes. Already in our earlier class, we have discussed prokaryotes and eukaryotes, but today in this class, we are going to discuss detail about prokaryotic cell and eukaryotic cell. So first, we are going to discuss about cell theory. So what is cell theory? Cell theory is the theory that is based on the fact that every living organism is made up of cell. So in this class, we are going to discuss about cell theory. The theory was put forwarded by Selidanen and Sivan in which year? In 1839. It states that the body of a cell, the body of an organism that are formed by cells and their products. And the cells are structural and functional unit of life. M.J. Selidan, the scientist who developed cell theory, M.J. Selidan in 1804 to 1881. It was a German botanist who published his findings in 1838. And he showed that there are, in case of plant tissue, they are made up of one or different types of cells. And the cells are unit of structures and tissues, and each cell has a broadly similar structure of cell wall, nucleus, and surrounding jelly-like structure that is called protoplasm. And Sivan, Theodor Sivan, from 1810 to 1882, that was also a German zoologist. That means the cell theory was proposed by one botanist that is called M.J. Selidan, and another zoologist that is called Theodor Sivan, who found that animal tissue were made up of cells and animal cells differ from plant cell in lacking cell wall. So after that, after the discovery of cells, the cell theory was proposed, and then the theory was not clear about the origin of new cells. So how the new cells arises from cells? That was not cleared by M.J. Selidan and Theodor Sivan. So it led to the formation of cell theory, but they believe that new cells are formed due to the cytoblast. That means it arises in form of a bud from the cells. But Rudolf Virchow conclusively proved in 1855, Rudolf Virchow proved that the new cells develop from pre-existing cells, and he called it as a omnicellula, cellulicellula, omnicellula. It is a Latin word, and its meaning the cell arises from pre-existing cells. So the cell theory has been modified, and later it defined as, according to new modern modification of cell theory, each body or every living organism is made up of cells and their products. Secondly, cell develops from pre-existing cells, and the cell theory are also defined that all cells are basically similar. They are made up of a piece of protoplasm. They consist of protoplasm and are enveloped by or surrounded by a thin membrane with or without cell wall. In case of animal cell, there is no cell wall. That means they contain the outer limit that is called cell membrane. And in case of plant cell, besides cell membrane, they contain cell wall. And next is the all cell possesses similar chemical composition and metabolic activities. And next, cells are not only unit of structure, but also unit of functions in living organisms. And they are the sum total of activities and interaction of cells. So these are the points. If you asked or if you have to write about cell theory, you first write who proposed cell theory and what is cell theory. Cell theory means it is the theory that elaborately described by M.J. Selidan and D. Sivan. According to them, all living organisms are made up of cell, and that is called cell theory. And then later, Rudolf Virchow proposed that new cell arises from pre-existing cell. And after that, it leads to the modification of cell theory into the following 5 points. You can write also more points, but here it is given 5 points. Then write this 5 point also. Next, here you see overall view of a cell. If you see the cell in microscope, then how we see it consists of an outer envelope, genetic material, and cytoplasm. What is outer envelope? Outer envelope means outer limit of the cell. Outer limit of the cell is the outer membrane that is called plasma lemma or plasma membrane. It is a living membrane. It is found both in plant cell and animal cell. But in case of plant cell, the outer limit is cell wall. Below the cell wall, plasma membrane is present. But in case of animal cell, the outer limit is plasma lemma or plasma membrane, not cell wall. Next is genetic material. Genetic material is DNA. In case of eukaryotic cell, it is found in the nucleus in form of what? In form of chromatin reticulum or chromatin material. In case of prokaryotic cell, it is called nucleoid. It is naked and it is found in circular form and found in the cytoplasm. Third is in case of cell, it is the cytoplasm. It is a semi-fluid matrix that accommodate the living materials, that is called cell organelles. Cell organelles, and they may be membranous cell organelles. Membraneless cell organelles are ribosome and centriole, and single membrane bound cell organelles are endoplasmic reticulum, Golgi complex, lysosome, microbodies, spherosome, etc. And double membrane bound structures or cell organelles are mitochondria and plastid. So these are the basic cell components found in a cell, whether it is prokaryotic cell or eukaryotic cell. Next is in case of prokaryotic cells are morphologically very primitive cell. It is a single membrane surround the cell. It is without membrane bound nucleus. That means nucleus is absent. And here in prokaryotic cell, the devoid of membrane bound cell organelles like plastid, mitochondria are absent, and advanced type flagella is absent. And they reproduce how? By asexual method. The asexual method means that is called binary fission or budding. In these two processes, they can reproduce. And for example, prokaryotic cells, that is bacteria, cyanobacteria, other name is called blue-green algae. Then mycoplasma, PPLO, shortly it is called PPLO, and its full form is pleuropneumonia-like organism. Next is in case of bacteria are two types. One is called gram-positive bacteria and other is called gram-negative bacteria. That means bacteria are divided on the basis of their staining capability. That means whether they can take acetocarmine, whether they can take the acetocarmine and or not. If the bacteria, they can be stained, they are called gram-positive, or they retain the purple color, that is called gram-positive bacteria. And if they can't retain the color of the stain, that is called gram-negative bacteria. So here you see the bacteria that are stained by gram staining. And for example, Diplococcus pneumonia is a gram-positive bacteria. The cell wall of gram-positive bacteria contain a polysaccharide and mucopeptide, as a result of which they can retain the purple color if you stain the particular bacteria. And in case of gram-negative bacteria, for example, Escherichia coli, its cell wall does not contain the mucopeptide or polysaccharide. So they cannot retain the stain, and it is called gram-negative bacteria. But they have in addition an outer layer of lipid, complex with protein and polysaccharide. So what is the difference between gram-positive and gram-negative? Gram-positive, they can retain the gram staining color, while gram-negative cannot. In gram-positive bacteria, the cell wall contain a polysaccharide and mucopeptide, while in case of gram-negative, the polysaccharide or mucopeptide layer is absent, but outer layer consists of lipid complex with protein and polysaccharide. Again, bacterial cell is covered by an outer slimy layer, a middle cell wall, and an inner cell membrane. In case of bacteria, some bacteria are very resistant. These bacteria always contain an outer slimy layer and a cell wall and an inner cell membrane. So this type of bacteria resistant bacteria. But in some cases, this slimy layer is absent. They are not resistant. So these are the basic difference between gram-positive and gram-negative and whether they are resistant bacteria or not. With the presence or absence of this slimy layer or capsule, we can say if capsule is present, it is resistant bacteria. If capsule is not, it is not resistant bacteria. So in our next slide, here you see the diagram of an bacteria. It is a prokaryotic cell. Here you see the capsular layer is present here. That means it is a resistant bacteria. The nuclear material is found, that is called nuclear, in the cytoplasm, and ribosome is present, that is called 70S ribosome. This bacteria is a motile bacteria. It can move with the help of a single flagella is present for movement. And there is some structure in the outer capsule layer that is called pili. That means one bacteria can identify another bacteria, whether it is male or female, by presence of these pili. These are sex pili. So this is the basic structure of a bacteria. Next is in case of eukaryotic cell, the cell wall present in case of plant cell. Plasma cell wall is dead, actually. It is rigid. It maintains the structure of the plant cell. Plasma membrane lie below the cell wall. It is a living semi-permeable membrane. It can allow water to move inside, but not the solid material come inside. Ribosome and inclusion body, these are found in eukaryotic cell and prokaryotic cell also. In prokaryote 70S, in case of eukaryote it is 80S. In case of bacteria, mesosome is present. Mesosome is not found in the eukaryotic cell. What is mesosome? In some bacteria, in which bacteria? Bacillus subtilis. It is a soil bacteria. The plasma membrane giving out certain invasions on infolding. That means the plasma membrane sometimes form folding like structure inside the cytoplasm. These are called mesosome and they help in respiration. Mesosome plays important role in separation of two daughter cells during the division, binary fission. From one cell, it forms two daughter cells by the help of mesosome. And genetic material, in case of bacteria, it is the circular. It is called nucleoid. And in case of some bacteria, the circular DNA, other DNA besides genetic material, there is also another DNA is present that is called plasmid. And the locomotory organ is present in bacteria that is called flagella. That means this part, the cell wall is present, ribosome and inclusion bodies, mesosome, these are all part of prokaryotic cell, not in eukaryotic cell actually. Okay. Next slide, here you see the eukaryotic cell. Eukaryotic cell, we know that it is the highly developed cell characterized by compartmentalization of cytoplasm through the presence of membrane-bound cell organelles. That means cytoplasm accommodates membrane-bound structure. Those are mitochondria, endoplasmic reticulum, Golgi complex, and a true nucleus is the important characteristic of eukaryotic cell. So this is the structure of eukaryotic cell. Here you see the mitochondria, microtubules, centrosome, microfilaments, lysosome, microfilaments, smooth endoplasmic reticulum, rough endoplasmic reticulum, peroxisome, cytoplasm, Golgi complex or Golgi apparatus, rough endoplasmic reticulum, ribosome, 80S ribosome, mitochondria, etc. present in eukaryotic cell. These are the animal cell. Here cell wall is absent. Next is the plant cell. Here you see the cell wall is present and the nucleus is well organized. Vacuole is also present. But here one important question is, what is the difference between plant cell and animal cell? So here you see in plant and animal cell, the common organelles are found. These are mitochondria present in both Golgi apparatus found in also both. Rough and smooth endoplasmic reticulum also found in both cells. Nucleus is well organized in both cells. Cytoplasm is also present, and ribosome here also ATS type in both cells. But what is the basic difference between plant and animal cell? In plant cell wall is present. Here, animal cell not present. In plant cell, large vacuole is present. In animal cell, small or no vacuoles. In plant cell, chloroplast is present. In animal cell, no chloroplast is present. In plant cell, flagella only in gamete. In plant, flagella is found only during the formation of gamete, which is the reproductive spores. And in case of animal, flagella normally present. So these are the basic difference between plant cell, animal cell, and what is prokaryotic and eukaryotic cell, and what is cell theory. So in today's class, we have discussed briefly about cell theory and the difference between plant and animal cell, and also the describe elaborately about prokaryotes, that is bacteria cells, etc. So I think students have got well this part. And in next class, we are also going to discuss about different cell organelles, textbook part. Okay, students, thank you for attending the class.